The overall goal of this procedure is to visualize the branching patterns of individual axons of DRG neurons in a whole mount preparation of mouse embryonic spinal cord in order to identify molecules involved in axonal branching. The projections of DRG neurons into the mouse spinal cord offer a simple, easy accessible experimental system to study axonal branching. When afferent axons of DRG neurons reach the dorsal root entry zone of the cord, they display a stereotyped pattern of T or Y-shaped bifurcation. The two resulting daughter axons then proceed in rostral or caudal directions respectively at the dorsolateral margin of the cord. Only after a waiting period, collaterals sprout from these stem axons to penetrate the grey matter and project to relay neurons in specific laminae of the spinal cord where terminal arborization occurs. This video demonstrates first how to dissect the spinal cord with its attached DRG from an embryonic day 12.5 mouse. Following fixation of the specimen, minute amounts of DiI are applied to the DRG using glass needles pulled from capillary tubes. After an incubation step, the labelled spinal cord is mounted as an inverted open book preparation to analyse individual axons using fluorescence microscopy.
The branching of axons is an important aspect in the development of neuronal connectivity. The method presented here allows a comparison of branching patterns of wild type with those of mutant mice, and thereby enables the identification of molecules that regulate axonal branching.
First,